The somatic pairs are derived from mesodermal tissue, from mesodermal cells, and give rise to uh, certain axial structures such as bone and muscle, particularly of the back area of the animal. Right. So in general, we can sum up neurulation as the creation of the longitudinal axis of the animal. All right. Let's take a look at how the phenomenon of neurulation can be appreciated in a gross perspective. Here we're seeing a uh, diagram, or we're seeing two pictures that represent the stages of neurulation, namely shaping, folding, elevation, and convergence. So you can say that the period of neurulation starts from the early traces of neural plate development until this neural plate later on transforms as an enclosed tube. The third group of cells, or the third group of tissue, derived from the general ectoderm, which is actually the neural tube. No? In a little while, we'll be talking about the process, okay, of primary neural induction, which includes all these four events, or all these five events, I'd say, if we're going to subdivide shaping and folding, or categorize shaping and folding as different procedures. Um, Alright. Before we examine the molecular component, or the molecular perspective, and the gross perspective, how the cells of the general ectoderm later on transform as neural tube, and the two other groups of the two other groups of tissues, neural crest and general, or skin cutaneous ectoderm. Okay, let's first talk about what induces the process of neurulation. In this diagram, we are seeing the cross, or the longitudinal section of an amphibian embryo. We've identified a very important structure in this image. If you remember the dorsal blastoporal lip, right? So you know that both the dorsal and the ventral side of the blastopore for amphibian embryos develop bottleneck cells that pull mesodermal endodermal fated cells interior of the developing animal. We expect that this dorsal blastoportal lip later on becomes the successor of the nucleus center cells. Later on becomes the Spearman organizer, which is important for mesodermal induction. In other vertebrates like mammals, avians, and reptilians, we are seeing a different structure that is homologous to the blastopore, the primitive streak. Expect the Hensen's node area serves as the anterior landmark of the animal, or the anterior landmark of the developing animal. Hensen's node serves as the entry point for chorda mesodermal cells. So we can deduce that both blastopore, the dorsal blastoporal lip, and the Hensen's node are important structures in the initiation of the process of neurulation, because they serve as entry points, or structures that allow the interiorization of chorda mesodermal cells. As they welcome chorda mesodermal cells that later on transform as notochord, the primary inductor for neurulation to take place. We expect the pattern to be from caudad to craniad, or caudad to cephalad. Chordal tissue that induce the overlying ectodermal cells actually start uh, from the Hensen's node, or the dorsal blastoporal lip, and then they push anterior, or they push towards the head portion of the animal.